Hello everyone, Mr. Linder here. Let's talk about the process of hematopoiesis. So hematopoiesis is the production of blood cells that primarily takes place in the bone marrow. So the production of blood cells actually has to begin with something called a stem cell. And so in our bone marrow, we essentially have what's known as a hematopoietic stem cell. That hematopoietic stem cell has the ability to differentiate into other types of cells. If you look at a diagram like this, what you see is that there are various pathways for the production of different types of blood cells. So for example, this lineage is for the production of red blood cells. So this would be known as erythropoiesis. If you look at this lineage here, this would be the production of platelets. So this would be known as thrombopoiesis. And then the remaining pathways, where you see the production of different types of white blood cells, this would be known as leukopoiesis. So erythropoiesis, thrombopoiesis, and leukopoiesis are actually just subdivisions of the concept of hematopoiesis. So hematopoiesis means the production of the different types of blood cells, and then we can focus in on individual production. So the production of red blood cells, erythropoiesis, the production of platelets, thrombopoiesis, and the production of white blood cells, which is leukopoiesis. Now, as we look at stem cell lineages, what we find out is that as these stem cells continue to differentiate, they eventually become essentially what we call progenitor type cells. And progenitor cells are committed to the cell type that you are producing within the bone marrow. So stem cells, especially the hematopoietic stem cell, that's definitely this sort of uncommitted stem cell lineage. As you differentiate and you become more and more committed, those cells will only have the potential to become certain types of cells. So it's actually rare to find uncommitted stem cells within human bone marrow. It's difficult to isolate and it's difficult to study because there are fewer and fewer uncommitted stem cells in the bone marrow. So an interesting technique that's been developed is to harvest umbilical cord blood. If you harvest umbilical cord blood and you store umbilical cord blood, you actually have access to hematopoietic stem cells that could be used by the donor down the line in the future. And so if the donor of the umbilical cord blood develops a cancer like leukemia, or they develop some other hematological disorder, you could take the stem cells that are in the umbilical cord blood and use those as a treatment option rather than looking for a donor and transplantation from somebody else that would be a match for that person. So being able to study and being able to have access to uncommitted stem cells is very important as we progress with biotechnology and treating disorders. The process of hematopoiesis, so if we were to sort of narrow in on what's going on, is really being able to look at the chemical signaling that affects the different types of stem cells so that we can produce red blood cells, platelets, and white blood cells. So on this particular diagram, what I want you guys to see also is the differentiation into two types of stem cell lines. There's the myeloid stem cells and then there's the lymphoid stem cells. You'll notice with the myeloid stem cells, you have the ability to produce progenitor type cells that are going to be able to develop red blood cells, be able to produce platelets, and then we're also going to be able to produce 4 of the 5 types of white blood cells. And so what we have is the myeloid lineage that's capable of producing a variety of types of cells within the bone marrow. The lymphoid lineage is designed to produce types of lymphocytes, so B lymphocytes, T lymphocytes, and then other varieties of cells like natural killer cells. So the lymphoid lineage is specific for the lymphocyte line. Myeloid stem cells are specific for other types of cells within the bone marrow. Before you actually, though, use bone marrow to produce blood cells, you actually needed to be able to produce blood cells in embryonic development. So essentially what's going on during about the third week of embryonic development, what you have is you have a specialized group of cells that are from the yolk sac of the embryo. And these specialized cells essentially form a cluster of endothelial blood vessels. So you have endothelial blood vessels. And these endothelial blood vessels that are developing embryonically are essentially the vessels that are going to initially produce blood. And in order to do that, there needs to be chemical signals. So this vascular endothelium is producing cytokines. Okay, so vascular endothelium is just referring to these endothelial blood vessels. They're producing cytokines. Cytokines are cellular chemicals that are essentially controlling hematopoiesis from this early stage of development. So at the third week of development, you're basically using a cluster of blood vessels to produce the chemical signals necessary to produce blood cells. As you continue to develop as an embryo, you're going to start to produce organs like the liver. You're going to see spleen development. And then, of course, you're going to start to have bones. And a part of bones is, of course, bone marrow. So rather than producing blood cells just from this vascular endothelium, you're going to start to see hematopoiesis in your embryonic liver, your embryonic spleen, and then, of course, your embryonic bone marrow. So as you are developing, you are seeing blood cell production taking place in other organs within the embryo. By birth, your liver and spleen essentially are no longer producing blood cells, so we can cross those out. Liver and spleen, by the time you're born, are not producing blood cells, and so we rely heavily on bone marrow. So during infancy, adolescence, and into adulthood, we're relying on bone marrow to go through the hematopoietic process and produce red blood cells, platelets, and white blood cells. As we age, our skeleton, essentially our adult skeleton, is losing active sites for blood cell production. So as you become an adult, the main places for blood cell production are going to be in the epiphyses of long bones, so that's the ends of long bones. Your ribs still produce blood cells, vertebrae still produce blood cells, pelvis still produces blood cells, and even portions of your cranium produce blood cells. So rather than all of your bones actively making blood cells, it's specific areas within the adult skeleton. The active portion of your bone marrow is known as red marrow. So we typically see, again, that red marrow in the epiphyses of long bones. So the epiphysis of a long bone is the ends of a long bone, and that's where you see the red marrow. And red marrow is where you're actively producing blood cells. The yellow marrow that's seen in bones is in the medullary cavity or marrow cavity. So the marrow cavity of a long bone will have yellow marrow, and that's filled essentially with adipocytes for fat storage. When you look at active bone marrow, what's interesting to see is that active bone marrow actually only has about 25% of the cells actively producing red blood cells. And then about 75% of the active marrow is being used to produce white blood cells. And that's interesting because there's actually a lot more red blood cells within your blood than there are white blood cells. But white blood cells have a much shorter half-life. Okay? So the life expectancy, if we look at the half-life of white blood cells have a half-life that is much shorter than the half-life of red blood cells. And so therefore we're constantly producing and replacing more of our white blood cells. So when you look at active marrow, you have to understand that you're actually seeing a larger percentage of the stem cell activity leaning towards the production of white blood cells than you actually do towards the production of red blood cells. And again, that has a lot to do with white blood cell function as it relates to immunity and protecting us against pathogens. So what are the different cytokines that are triggering these hematopoietic processes? So what's triggering erythropoiesis, thrombopoiesis, and leukopoiesis? The cytokines, and we're going to focus on the predominating ones. There are lots of cytokine chemicals and chemical signals. But when you're focusing on the main chemicals, we want to focus on EPO. EPO stands for erythropoietin. Erythropoietin is a hormone, a cytokine chemical, but it's classified as a hormone that is released by our kidneys to trigger red blood cell production. Thrombopoietin, TPO, is a cytokine chemical that is produced by the liver to influence platelet production. And then our main chemicals for producing white blood cells are referred to as CSFs, or colony stimulating factors. There are other chemicals necessary for white blood cell production. So there are interleukins as well. A variety of interleukins play a role in hematopoiesis. Interleukins are also vitally important for immune system function as well. But these interleukins are chemical signals that are used to communicate between white blood cells. So interleukins. And there are a number of interleukins. So we call them interleukin 1, interleukin 3, interleukin 7. We number the interleukins, and these are communication signals between white blood cells. So between interleukins and colony stimulating factors, we can use these chemical signals to produce the various types of white blood cells. Let's take a closer look at leukopoiesis. So if we were just going to focus on the production of white blood cells, what we call leukopoiesis, then we know we're focusing on this idea of colony stimulating factors. What you should note, again, is that it takes both myeloid stem cells and lymphoid stem cells to produce the 5 types of white blood cells. So in order for us to make basophils, eosinophils, neutrophils, monocytes, and lymphocytes, we're looking at both of these stem cell lines. The most important colony stimulating factors would be the G-CSF colony stimulating factor and the GM-CSF. But again, there are a variety of interleukins and CSFs for producing the different types of white blood cells. What do colony-stimulating factors do? They essentially trigger mitosis, and they're triggering these stem cell maturations so that you can make leukocytes. So the GCSF line, if you follow that lineage, GCSF is the lineage that gives us neutrophils. And it turns out that neutrophils are the most abundant white blood cell that we have. So neutrophils are the most abundant. Basophils are the least abundant. So there's a hierarchy, if you will, of how many white blood cells we actually produce in our bone marrow. So GCSF is important because it's producing the most abundant white blood cell. And neutrophils are highly important for fighting bacterial type infections, which are things that could happen on a daily basis as bacteria invades our barriers of the body. We need to make sure that neutrophils are getting to those locations and fighting against those particular pathogens. GM-CSFs, so this colony-stimulating factor, is important for the production of the monocyte lineage. And it turns out that monocytes are the precursors to macrophages. So this is an important lineage for white blood cells because this is where we get our macrophage cells from, and macrophages obviously are involved in things like phagocytosis. So again, they're helping us in the immune system to fight off a variety of pathogens. Monocytes are the third most abundant white blood cells. So neutrophils are first, followed by lymphocytes, then followed by monocytes. So it's a very important lineage of white blood cells in terms of immunity for the body. All of the white blood cells, though, obviously have a variety of roles. We know that lymphocytes are vitally important to combating predominantly viral infections. You've probably heard a lot about eosinophils and their ability to fight parasitic worm infections. And so clinically understanding what the white blood cells do, being able to do differential white blood cell counts so that we can determine what type of white blood cell is elevated in the blood can all be used in a diagnostic way to figure out what's going on if somebody has an illness. What's interesting about all of these cytokines, the CSFs, the TPOs, the EPOs, these have all been essentially bioengineered. They've been cloned, and we're able to actually produce synthetic versions of these chemicals so that you can actually increase blood cell production. So there are EPO drugs out there, there are TPO drugs out there, and there are even colony stimulating factors that can be used to stimulate bone marrow to make the different types of blood cells. So again, to recap the chemicals, we have colony stimulating factors and interleukins, which are used in leukopoiesis, so being able to produce white blood cells. The thrombopoietin, again, is produced by the liver, and this is going to trigger megakaryocytes. And megakaryocytes are the precursor in bone marrow to platelets. So megakaryocytes fragment and therefore give us platelets that we use in the hemostasis process, so fixing broken blood vessels. And then erythropoietin is the hormone from the kidney that is responsible for erythropoiesis. So I want to focus just really quickly on that last portion here. So this is a pathway, if we were just to zoom in on the pathway for red blood cell production. So this is looking at erythropoiesis. And so looking at erythropoiesis, the production of just red blood cells. Again, hematopoiesis is making all of the blood cells. Erythropoiesis focuses on red blood cell production. So the hormone is erythropoietin, that's EPO. And the reason that EPO is produced by the kidney is in response to hypoxia. So hypoxia is a decrease in the oxygen carrying levels within the blood. So when you are hypoxic, you essentially have low levels of oxygen. And that becomes a stimulus for secretion of EPO. So when a human becomes hypoxic, we release EPO. EPO becomes a trigger for the production of red blood cells. How does that actually work? Well, the low levels of oxygen are actually triggering a gene, okay? So it's basically what's known as hypoxia-inducible factor. And that hypoxia-inducible factor is then, that gene is triggered to be transcribed. So we have what's called transcription that takes place, and then we have translation of the EPO hormone, and then that EPO hormone can be released by the kidney to trigger the bone marrow. So what's happening? When EPO triggers the bone marrow, that's then triggering this cascade of effects in order to produce red blood cells. So EPO is triggering the stem cells and these progenitor cells to go through their production. So proerythroblast to erythroblast to normoblast, and then eventually leaving the bone marrow, expelling the nucleus, getting rid of the organelles, and becoming a reticulocyte, and then maturing into a fully functional red blood cell. What essentially is a fully functional red blood cell? It's a plasma membrane filled with hemoglobin, so HB is hemoglobin, and hemoglobin carries oxygen. And so that brings us back to why the stimulus for release is hypoxia, right? Low levels of oxygen is going to trigger the production of more red blood cells so that you have more hemoglobin and you can carry more oxygen, thus increasing your oxygen carrying capacity. So what we've discussed is hematopoiesis, looking at erythropoiesis, thrombopoiesis, and leukopoiesis, and that is the production of blood cells in our bone marrow. I hope that helps. Take care.